In this part, we discuss about the gametogenesis. So what is gametogenesis? Formation of gametes. In case of male, that is spermatogenesis, formation of sperm cells. And this spermatogenesis takes place in testes, primary sex organ. And in case of female, that is formation of egg cell, that is called as oogenesis, and oogenesis takes place in ovary. Now first we discuss about the spermatogenesis. So spermatogenesis takes place in testes. Testes contain seminiferous tubules, and seminiferous tubules have a germinal epithelium. These germinal epithelium are diploid in nature. Okay, so this germinal epithelium shows first phase, that is called as multiplication phase, through which these germinal cells are divided mitotically. Okay, after the mitotic division, it produces the first cell, that is called as spermatogonia. Now this spermatogonia also diploid in nature. Some spermatogonia are act as type A, and which is act as the stem cells which divide continuously mitotically, and this is act as the stem cells, while some spermatogonia are showing the further growth phase, and from that sperm cells are producers. So type B spermatogonia, which shows the next phase, that is called as growth phase, in which the size of spermatogonia is enlarges because of nutrition. So it becomes the primary spermatocytes. From spermatogonia, primary spermatocytes are producers. We just take one example. So primary spermatocyte, which is also diploid in nature. Now this primary spermatocytes shows the maturation phase, and during this maturation phase, primary spermatocyte shows meiosis. So meiosis is called as reductional division, and meiosis takes place by two phases, that is meiosis 1 and meiosis 2. Reductional division, here the chromosome number is reduces. For example, in case of primary spermatocyte, 46 chromosomes are present, but after this meiosis, whatever the spermatid cell produces, in which 23 chromosomes are present. Okay, so meiosis 1, primary spermatocyte shows the reductional division, that is called as meiosis. Meiosis 1. After meiosis 1, two cells are produced, that is called as secondary spermatocytes, and these secondary spermatocytes are haploid in nature. Only one set of chromosome is present in secondary spermatocytes. This is very important concept. So primary spermatocyte shows meiosis 1, through which secondary spermatocytes are produced, which are haploid in nature. Now this secondary spermatocytes also shows next meiosis. Meiosis 1 and then meiosis 2. Secondary spermatocyte shows this meiosis 2, meiosis 2. After the meiosis 2, 4 haploid cells are produced, and that 4 haploid cells are called as spermatids. So this is the spermatids. Finally, spermatids are produced from this germinal epithelium. Okay, you understand this? So first, germinal epithelium shows the first phase, that is called as multiplication phase. Through it just mitotically divide and conversion into spermatogonia. Okay, and then this spermatogonia shows second phase, that is called as growth phase, through which primary spermatocytes are produced. So these primary spermatocytes are also diploid, and this primary spermatocyte shows the third phase, that is maturation phase, through which primary spermatocytes divide meiotically, and then secondary spermatocytes, and finally 4 spermatids are produced. Okay, but these spermatids are not like a mature sperms. Its shape is different, it is inactive. Okay, so from this inactive spermatids, active sperms are produced, and that process is called as spermiogenesis. So spermiogenesis means formation of active sperms from spermatids. So what changes takes place during this spermiogenesis? So because of that, these inactive spermatids convert into the sperms. So first change takes place in this spermatid, that is condensation of nucleus, in which loses of mRNA from that spermatids, mRNA and acidic proteins are loose, only DNA molecules are remaining into the basic proteins and DNA molecules are remain into the nucleus of spermatids. So first change in spermatids for the formation of sperms, that is condensation of nucleus. Then next is pointed, its shape is changed. So here its shape is just a circular, but actual shape of sperm is like this. So its shape is pointed, anteriorly oval and flat in case of human. So next is change occur in the Golgi body. Okay, so Golgi body is divided into two parts, that is larger part called as acrosomes, which play very important role, which is present in upper side of the head, and that plays important role. It secretes sperm lysines. Okay, it secretes some enzymes. Then smaller part, that is Golgi rest, which are degenerated. So Golgi body shows changes during this spermiogenesis, that is conversion of this Golgi body into that divide into two parts, that is large and small. Large is called as acrosome, and acrosome secrete enzymes, and this small part is called as Golgi rest, which are degenerated. Then this complete sperms is now covered with plasma membrane. Okay, so covering is present from this head part to tail part, but cytoplasm is very less. Reduction in cytoplasm, there is no cytoplasm present in the sperm cells, only cytoplasm is present only in tail region. Okay, except tail region, there is no cytoplasm present in sperm cells. So it is covered with plasma membrane and cytoplasm present only in the tail region. Okay, and one more important change occur in spermatids, that is mitochondria. The organelle mitochondria is changed. It becomes shows the spiral structure, and that spiral structure is called as Nebenkel, and this is very important for the motility of sperm, for energy production. Okay, so mitochondria are converted into spiral structure, then this is called as Nebenkel. And then centriole, at which placed at the neck region of the sperm, that is that distal centriole, proximal and distal centriole are placed at the neck region. These are the some changes occur during spermiogenesis. Spermiogenesis means formation of active sperms from inactive spermatids. So formation of sperms from spermatids, that process is called as spermiogenesis. Okay, so condensation of nucleus, its shape is changed, Golgi body, changes occur in Golgi body, then this complete sperms is covered with a plasma membrane, then cytoplasm present only at the tail region, and mitochondria converts, that shape of mitochondria is changed, that is spiral structure, and then Nebenkel, that is called as a Nebenkel, and centriole, proximal and distal centriole at the neck region. So this is the spermiogenesis and spermiation. One more term is there. Spermatogenesis means formation of sperm cells, that is spermatids, finally spermatid formation. Then complete process is called as spermatogenesis. Spermiogenesis means formation of sperm cells from spermatid, and spermiation. So Now we discuss the structure of mature human sperms. Sperm cells are haploid, flagellated, motile and microscopic cells. So it is divided basically into four parts, that is head part, then neck part, middle piece and tail region. Okay, so the head part having two main structures, that is acrosome and nucleus. Acrosomes are produced from Golgi complex. So Golgi body is divided into two parts, that is Golgi that is larger part is acrosome and Golgi rest. So this acrosome produces enzymes which helps to break the membrane near the egg cell. Okay, so and this is the oval shaped nucleus. So now this is covered by plasma membrane. The head part is covered by plasma membrane, and it also have a special type of covering other than this plasma membrane, that is called as galea. Okay, so this is the covering, this is the head part, and it having very small depression, and that is called as neck region. And this depression part contains two centrioles, that is the proximal centriole which is present in the depression of nucleus, and the second centriole is present in the middle piece region. So this centriole present in near the nucleus, that is called as proximal centriole. One centriole which is present in the depression of nucleus, that is proximal, and this proximal centriole is essential for the spindle formation during first zygotic division. After the formation of zygote, after fertilization, zygote is produced. During this zygotic division, this proximal centriole help for the spindle formation, for mitotic division. So the proximal centriole, and the next is the distal centriole. This distal centriole forms axial filament, or that is called as acronym. Okay, so which is 9+2 microtubules, while this proximal centriole is 9+0 microtubules. So proximal centriole 9+0 arrangement, and in axial filament 9+2 arrangement of microtubules. So then this middle piece, neck, is very short depression which is present near the nucleus. Now middle piece also covers by plasma membrane, and also one more covering is there, that after this plasma membrane the covering is that called as manchete, and it contains, that is axoneme also, and mitochondria, and that mitochondria are spirally arranged. We call them as a Nebenken. Mitochondria are spirally arranged in middle piece. Okay, so this middle piece part contains axoneme and this Nebenken. Nebenken means spirally arranged mitochondria. It also have, so this is a special covering which is from the proximal or distal region. So lower region of nucleus, it starts from lower region of nucleus till the middle piece, and that covering is called as manchete. Okay, so this is the special covering which is from the end part of nucleus to the middle piece. Okay, and Nebenken means spirally arranged mitochondria. The next is the tail part. Tail part is containing cytoplasm. So cytoplasm is not present overall this sperm cells, it is present at the end, that is that is the end piece part, and end piece also not having plasma membrane. So end piece, it's having very long flagellar structure, tail is a very long structure, and at the end piece there is no plasma membrane covering. Okay, so this Nebenken helpful for production of energy. Okay, now we see that in case of male, spermatids are produced from this secondary spermatocytes. 4 cells are produced. From that, one contains the X chromosome and another contains the Y chromosome. So this secondary spermatocytes divided into 4 spermatids. So this is the X primary secondary spermatocytes and Y secondary spermatocytes. So now this produces 2 spermatids and Y containing chromosome produces 2 spermatids. So like this division is there. So 2 sperms that contains the X chromosomes are produces, while the 2 sperms are containing Y chromosomes. Okay, so X containing are called as gynosperms and Y containing sperms are called as androsperms. So from this 4 cells, 4 sperms are produces. That 4 sperms, out of that 2 are X containing and 2 are Y containing. X containing sperms are called as, X chromosome containing sperms are called as gynosperms, while the Y containing sperms are called as androsperms. So this is completely depend on male what is the sex of fetus, because female having only X chromosome. Okay, so in case of male, that is X and Y chromosome. 50-50% chances of X chromosome or Y chromosomes. So gynosperms, X containing chromosome, X chromosome containing sperm, and Y, that is androsperm, that means Y chromosome containing sperm. Hello students, now we discuss about the oogenesis process. In previous videos we discussed about the spermatogenesis process, that means formation of male gamete. Now we discuss about the oogenesis, that means formation of female gamete, that is egg or ovum. Okay, and from primary germinal cells. This egg or ovum is produces from primary germinal cells under the influence of gonadotropic hormone like LH and FSH, and all this process takes place in ovary. Okay, so ovary is a primary sex organ in case of female. So oogenesis means the process of formation of female gamete, that is egg or ovum, from diploid germinal cells under the influence of gonadotropic hormones such as LH and FSH. So this is oogenesis, and it starts in females even before birth. In spermatogenesis, the process starts in puberty and then it is continuous. But in case of female, this process is discontinuous process. So it starts in females even before the birth. Okay, so basically it is divided into 2, that is some process takes place before birth and some process takes place after birth. So it is divided into 3 phases, that is multiplication phase, through which these diploid germ cells divide mitotically, that is multiplication phase, its number is increases. Then growth phase, the oogonia are developed into primary spermatocytes. Vitalogenesis process we'll discuss later. Okay, that is growth phase, and then the maturation phase. So this meiosis takes place during this maturation phase. Now multiplication phase. So these primary germ cells are produces in female fetus from endoderm layer of yolk sac. Okay, millions of copies of these germ cells are produces, and it shows a mitosis. Number of copies are produces again. So this produces oogonia. These diploid cells are called as oogonia. And first step is a multiplication phase, in which primary germ cell shows mitosis, through which million copies of this oogonia are produces. Now this oogonia shows a growth phase, and this growth phase occurs because of vitalogenesis, that means formation of yolk. Okay, and the size of oogonia is increases. Yes, in case of human female, the size of oogonia, this primary oocyte, is increases 200 times. Okay, because of yolk. Okay, so the primary oocyte, after this oogonia, in growth phase, oogonia convert into the primary oocyte. This is also a diploid cell. Okay, and primary oocyte starts meiosis 1, but the meiosis 1 is not completed. Meiosis 1 is arrested in diploteny phase of prophase. Diploteny phase of prophase 1. Okay, so it is arrested in meiosis 1, diploteny phase of prophase 1 of meiosis 1. So understand this, diploteny phase, it is arrested in meiosis 1, and this all process takes place before birth. So in human fetus, this primary oocyte are produces. This primary oocyte produces what? Meiosis 1, but it is arrested. Okay, arrested in diploteny phase. Okay, so this all process takes place before birth. Now, after birth, that means after 10 or 12 years, this primary oocyte number is decreases, and now after puberty, only few thousands of this oocyte are remaining. 40 to 60,000 oocyte are remaining after birth, and then puberty 10 to 12 years. Then under the influence of hormones, these primary oocyte complete meiosis 1, which are arrested in diploteny phase of prophase 1. This is completed after birth at the puberty. So the first, because of menstrual cycle, from all this oocyte, only one of it per month is developed into the secondary oocyte. Okay, so and this secondary oocyte is releases from Graafian follicle at the time of ovulation. So here we discuss it again. So primary oocyte, which is arrested in meiosis 1. Okay, then this meiosis 1 is completed when? It is not completed before birth or after birth. It is completed after, at the age of puberty. Okay, 10 to 12 years after the birth. Okay, now this is because of influence of hormones. So the primary oocyte completed its meiosis 1, and then it develops into the two unequal cells, that is secondary oocyte and first polar body. Remember that here nuclear division is equal, but cytoplasmic division is unequal. That's why size of secondary oocyte is bigger as compared to the polar bodies. So all this primary oocyte shows this or form this secondary oocyte? No. From out of 500 primary oocyte, only one of primary oocyte is developed and convert into secondary oocyte. Okay, at the start of menarche, and every month this one primary oocyte is. Every month only one primary oocyte is developed into the secondary oocyte. It shows the meiosis 1. It shows the completion of meiosis 1, and then it releases at the phase of ovulation, that is in the form of secondary oocyte. One bigger is a secondary oocyte and one small cell, that is called as first polar body. Why the size is different? Because it shows the equal nuclear division, but it shows unequal cytoplasmic division. Understand this? Okay. So this secondary oocyte and first polar body are produces after completion of meiosis 1, and this meiosis 1 is complete at the age of puberty. It starts from at the age of puberty. So from 500 of this primary oocyte, only one from per month, near about 40,000 to 60,000. यामध्ये किती आहे? 40,000 to 60,000 प्रायमरी oocyte आहेत, पण त्यापैकी 500 जे काही प्रायमरी oocyte आहेत त्यातून एकच पर मंथ एकच काय होईल? सेकंडरी oocyte मध्ये कन्वर्ट होईल. Okay, it complete its meiosis 1 and then it produces secondary oocyte and this polar body. So in menstrual cycle at the day of 14, this Graafian follicle is ruptured and secondary oocyte is releases. Okay, so this process is takes place before fertilization. Now, the secondary oocyte is released from ovary and the second meiosis is not takes place yet. So second meiosis takes place only when sperms are entered into the female vagina. So when sperms are entered, fertilization takes place. At that time only this secondary oocyte is divided, or secondary oocyte shows this meiosis second. Till that this secondary oocyte is arrested in metaphase of meiosis second. It is arrested in metaphase of metaphase. Okay, meiosis second is not completed. It will complete when there is an entry of male gamete, fertilization takes place. At that time this metaphase second is completed and this convert into the true female gamete, that is ovum and second polar body. So understand this, every time it is not possible, म्हणजे प्रत्येक वेळेस fertilization होईल असं नाही. So secondary oocyte is not converted into this ovum. Every time it is not converted into ovum. One or two time or three time in case of female life this is converted. The secondary oocyte is converted into the ovum at the time of fertilization, when sperm cells are entered and then this ovum is fertilizes. Okay, and then fertilization process takes place. So understand this. So the secondary oocyte enters into the meiosis second, but it is not completed. Okay, it complete when that is sperm cells enter and fertilization process takes place. After this fertilization process takes place. So this is overall oogenesis process. थोड़ा सा complicated आहे, but it understand by only some process takes place before birth and some process takes place after birth. So before birth which processes? Multiplication phase. In multiplication phase mitosis takes place. From that primary germ cells to oogonia are produced. This is also diploid cells, and then this oogonia are converted into the primary oocyte and that process is called as growth phase. Before birth this processes takes place. Meiosis 1 is also, but it is not completed. Okay, so meiosis 1 is complete at the puberty. Okay, so it is arrested in meiosis 1 and which phase? Diploteny phase of prophase 1 of meiosis 1, and then at puberty age, that is 10 to 12 age, it is start to develop. Not every oocyte, so some of that oocyte starts to develop and meiosis 1 is completed. When meiosis 1 is completed, then two types of cells are produced. One is a larger cell and one is a smaller cell. That larger cell is called as secondary oocyte and smaller cell is called as first polar body. Here equal nuclear division takes place but unequal cytoplasmic distribution. Okay, because of that one cell is larger and one cell is smaller. So this secondary oocyte is releases, yes, at the time of what? Fertilization, at the time of ovulation. Okay, so Graafian follicle ruptures and this secondary oocyte is releases. Now every time this secondary oocyte is not converted into the ovum, meiosis second is not completed in every secondary oocyte. Okay, so secondary oocyte starts to meiosis second but it is arrested, it is not completed. It is arrested in which phase? It is arrested in metaphase. Now if fertilization will take place, that means sperms are entered into this fallopian tube and at that time only this secondary oocyte is converted into the true female gamete, that is ovum. Okay, and again one more polar body is produces. Now we discuss the structure of secondary oocyte or ovum or female gamete, which is releases after the ovulation. Okay, so here only meiosis 1 is takes place.
So this is basically the secondary oocyte. The membrane, first membrane of this secondary oocyte is called as vitelline membrane. So inside of this vitelline membrane, nucleus is present and that cytoplasm is present, that is called as ooplasm. Okay, and after this vitelline membrane, one more layer, one more membrane is present, that is called as zona pellucida. One more covering, that is zona pellucida, which is produces from hard covering, carbohydrate or glycoprotein. It is the glycoprotein layer, that is zona pellucida. And space present between this vitelline membrane and zona pellucida is called as pre-vitelline space. So what is the pre-vitelline space? It is the space present between, this is vitelline membrane, and zona pellucida is called as pre-vitelline space. And inside of this pre-vitelline space, polar body, the first polar body which is produces by meiosis 1 is present here. Okay, the polar body present in pre-vitelline space. Okay. Outside of this zona pellucida, there is layer, one more layer, that is produces from glomerulosa cells called as corona radiata, and this corona radiata contains hyaluronic acid. So hyaluronic acid is present between this corona radiata layer. So because of that, they form a strong structure. It is not possible for any molecule, any substance or any cell enter into this because of this hyaluronic acid. If sperm cells want to entry into this, so they are not able to pass this layer. But sperm's acrosome produces some enzymes, that is one more enzyme of that, that is hyaluronidase, which breaks or which hydrolyzes that hyaluronic acids, and then only this corona radiata layer is separated, and then sperms get entry into this secondary oocyte. So understand this, corona radiata, in between the layer of this corona radiata, hyaluronic acid is present. If sperm wants to entry into this secondary oocyte, so or egg cell, so they break this or they hydrolyze this hyaluronic acid layer, and then they enter into this egg cell. Okay, so and this secondary oocyte shows polarity. Where the polar body is present or nucleus is present, that end is called as animal pole, and opposite of this, that is called as vegetative pole. So understand this, this is rounded and non-motile structure, and it is the largest cell in human body, human female body, that is 100 micrometer in size, that is 0.1 mm. Okay, its size is 100 micrometer. So this is the secondary oocyte which is releases after meiosis 1. So this layer is, first layer is called as vitelline membrane. Vitelline membrane contain nucleus and cytoplasm, that is ooplasm. And the second layer is called as zona pellucida, and space present between this vitelline membrane and zona pellucida is called as pre-vitelline space. And what is present in that pre-vitelline space? The first polar body is present in that pre-vitelline space. And outside of this zona pellucida, one more layer, that is called as corona radiata, and corona radiata contains or hyaluronic acid present between this corona radiata, and if sperms wants to entry into this egg cell, they break this corona radiata layer. So for that, acrosome of sperm cells produces enzyme, that is hyaluronidase, which breaks this hyaluronic acid, and through which sperms get entry into the egg cell. So it shows the polarity, that is animal pole and vegetal pole. Where the polar body is present, that is animal pole, and opposite is called as vegetative pole. Thank you.